I'm Ruba, and together with Param and Anisha, we've been working with a team dedicated to improving the impact and efficacy of mRNA therapeutic development. Our role was to assist in refining the market understanding and competitor landscape, ensuring our approach aligned with current needs. To this end, we conducted a detailed competitor analysis, identifying areas where our work could provide distinct advantages. We also researched emerging trends and applications to help the team flesh out the opportunity in this space. In the end, our contributions solidify the team's value proposition, and we are so grateful for the opportunity to learn more about translating laboratory research into commercialization. I'll now hand it over to our project team lead, Dr. Ilya Finkelstein.
Hello. I think we've all seen the power of RNA a few years ago, right? We all lived through a pandemic that was caused by 30,000 bases of RNA. And RNA was also our salvation. RNA vaccines saved us from a pandemic, and the numbers for the opportunity space here for RNA vaccines and therapeutics are staggering. Let's see, $73 billion market by 2030 for RNA vaccines and therapeutics. Over 13 billion doses of RNA molecules have been administered to people globally, with 70% of all people in the world having been injected with an RNA molecule. And this goes beyond just vaccines for COVID. This, of course, includes cancer vaccines and RNAs for therapeutics, including protein replacements, cancer therapies, et cetera. Massive market. Now, the interesting thing is all of this RNA has to be made in a test tube before it's deployed into humans. And it's made by one legacy enzyme, RNA polymerase made from a bacterial virus called T7. Now, this RNA polymerase makes a lot of mistakes. These mistakes cause cells to go into innate immune response, die, and actually cause systemic trouble for the patient. Our goal here is to develop a new next-generation RNA polymerase that can produce ultra-pure RNAs and enable RNA technologies like circular RNAs and super long RNAs for self-amplifying vaccines and other therapies. So that's our goal. We want to avoid the side effects, make long RNAs and circular RNAs, and produce ultra-pure therapeutics based on RNA, which is an emerging platform technology. Our approach combines mining the tree of life for completely new RNA polymerase variants from extremophile organisms that have learned a lot of tricks about surviving in extreme environments. Our lead candidates are incredibly specific, at least twofold, if not fivefold better than current technologies. And our goal now is to use machine learning technologies to further engineer them, improve these properties, and provide a drop-in replacement for existing T7 RNA polymerase. That will be our product. Our team consists of myself. I have over 15 years of experience in this space. Alexis Hill, who's a bioinformatician machine learning specialist, Dr. Salman Sidin, who's a protein engineer, and RNA transcription specialist. We have a phenomenal team that have really helped us with their enthusiastic support, market fit advice, and shaping this proposal towards a good product fit. So with that, our approach is as follows. We have lead candidates. We're optimizing them now. We're going to file IP on the designs within 6 months. Then we're going to refine these approaches, produce really compelling preliminary results that we're going to take to industry partners. We have quite a few industry partners already, conduct immunogenicity studies, and license this out globally. Now, we're already partnered with at least one company that produces mRNAs for R&D applications, and have already started talks with many others. And with that, I want to leave you and open time for questions for our approach.
Great presentation, thank you. How will your product be regulated?
So this is going to be, it depends on the market that it approaches. It's a pretty broad platform technology. For R&D applications, we're going to sell the enzyme either to enzyme manufacturers or people who make RNA, and they'll sell the R&D product to industry and academia. For GMP manufacturing, let's say for therapeutics, that's up to the pharma companies, right? So they're going to use this in a clean manufacturing, good manufacturing practices, so that then they can make sure that it meets all the safety and efficacy concerns that would be up to the regulators, if that makes sense. This is underlying all of that. And we're not selling the protein, we're selling, or they will be selling the RNA that is produced by the protein.
Hi. So you said that there are many markets out there.
Yeah.
Where do you see essentially the first market, the core markets that's going to actually launch this product or platform?
Yeah, thanks for that question. I think the fastest and shall I say easiest entry point is to manufacturers of mRNAs. So there's a lot of companies that you can go buy an RNA molecule from, and they're always looking for ways to make it faster, cheaper, and have capabilities that others cannot do with their legacy enzymes. So they're very interested. They want our stuff. So we're already talking to them because you don't have to go through all the regulatory hurdles of a in human use therapeutic, right, for R&D. And then after that, it would go into the therapeutics. Yes. That's right. We're going to file after we do the further optimization and mutagenesis to improve the properties of the enzymes. Yes, we've performed an IP analysis. I think our differentiator is twofold. A lot of people are currently trying to engineer T7 RNA polymerase itself. Uh-oh, are we out of time? I'm happy to discuss afterwards. Thank you for your question.